want to thank Gay and all you guys to sponsor Alice's trip. That makes us to be able to invite her here to give a talk. And so it's my great honor to introduce you our today's speaker, Dr. Alice Altman. And Alice got her PhD at University of British Columbia in oceanography. And after that, she moved to Montana State University, did a postdoc. And she worked a few years at the University of South Alabama before she moved to the Bedford Institute of Oceanography as a research scientist. And Alice's expertise is in microbial ecology. She did a lot of work in different types of ecosystems, including estuaries, wetlands, also hydrothermal vents, I believe. And she also published a lot of work in biodegradation, especially on hydrocarbons. And today she's going to tell us more about her work on biodegradation of hydrocarbon. And let's welcome Alice.
Okay. Thanks very much for coming. This is a much better turnout than I had for my last talk. I had pretty busy. I'm going to introduce you to a type of oil product that you may not be familiar with. So if anybody has done work in the Gulf of Mexico with the Macondo 252 oil, this is something totally different. Macondo 252 is a wonderful light crude that's easy to work with. Diluted bitumen is what we get when we have oil sands. And so I work for the Center for Offshore Oil, Gas, and Energy Research, which is part of the Department of Fisheries and Oceans up in Canada.
Is she a singer?
Yep.
See?
Maybe. Okay, so I just wanted to introduce you to COOGER, as we call it. And this is a research group that was established in 2002 to coordinate the research across Canada in oil spill understanding. And the two main points that they had was that they wanted to increase our knowledge about fate and behavior of oil in coastal environments as well as marine environments. And initially, this was really set up to understand how oil might behave if it was spilt off the coast of Newfoundland in the Hibernia oil fields region. But as you see, I put freshwater in there because that's the direction that a lot of our research is moving, is into understanding the interactions in freshwater. And we're also wanting to use this knowledge to understand and improve the effectiveness of mitigation measures, so whether those are physical mitigation measures or chemical mitigation measures or biological, so that we can better prepare and then clean up and respond to oil spills. And so recently, our focus has really been on bitumen and diluted bitumen. And so this is actually a picture of what the tar sands look like before they're, basically when they're pulled out of the ground. And so this is extracted from the oil sands in the Alberta, Saskatchewan area. It is classified as a non-conventional oil product. I mean, it's definitely heavier than a heavy oil. It is essentially a solid. And in order to transport this stuff, we need to actually dilute it so that we can decrease its viscosity. So diluted bitumen can be transported. There's two main ways that we can transport this product across pretty large distances, because Alberta and Saskatchewan are, for those of you who might not know the geography of Canada, just sort of north of Idaho and Montana area. And so we can use pipelines or we can use train systems. So pipelines, there's two different kinds of diluted bitumen products. One is called dilbit, where it's diluted with a light oil, usually something like a natural gas condensate. And then there's synbit, which is where they use a synthetic crude. For both of these, we have about 30% of the final product is actually this diluent, and the other 70% would be our bitumen. Challenge here is that each company actually has some sort of proprietary formula that they use for their diluent, and they actually have a little bit of changes between summer and winter, because in winter they need to have a different type of diluent so that it will still move through the pipelines when it's, you know, minus 40 Celsius outside. If we're looking at train, this is where we call it rail bit. It's essentially the same concept. It's just with trains they actually don't use as much diluent. It tends to be a thicker product. And there are some instances where you can move raw bitumen if you're going to use heated pipes or heated trains. That is a whole other sort of engineering issue that has to be dealt with. So I'm going to be talking about some experiments and work that we've mostly done with dilbit. And we've mostly been using 2 different dilbits, so I'm going to kind of show you data from both. One is called AWB, which is Access Western Blend, and that comes from the Athabasca region. And then there's a Cold Lake Blend, or CLB, and that comes from the Cold Lake region. And what the different regions mean that we have very different chemical compositions in our bitumen, and so we have different behavior in our dilbit. So this gives you an idea of the problem and why we have to dilute bitumen. So this is viscosity on a log scale of these 2 different types of bitumen. And you can see that we're up in the sort of 500,000 range. Arabian Light and South Louisiana Crude kind of fall down in this area, so we're now down to 10. And then once we actually dilute the bitumen, so these are those 2 different blends, you can see that they've reduced the viscosity quite a bit. So this stuff will flow, but it does not flow easily. It's really hard to pour. Setting up experiments and getting exact amounts added to our incubations is a challenge that we're constantly working with. So one of the reasons that we're really interested in this, and this is probably becoming more and more of a clear issue, is that there are a lot of proposals out there for pipelines, and there are a lot of pipelines in North America. And so what we have here is the solid lines are pipelines that actually exist already today, and the dashed lines are some of the proposed pipelines. So what we have here is this was called the Northern Gateway Pipeline. The idea was that the pipeline was going to go from the Alaska oil sands up here and would be loaded onto tankers up in Kitimat. And so that one actually in November was deemed not going to happen. However, the Kinder Morgan Trans-Canada pipeline was approved. So it was, I think, a little bit of a trade-off. And so what this pipeline is actually going to do is twin an existing pipeline and go into basically the Port of Vancouver. Keystone XL, big question mark there. So it's been approved on the Canadian side. It was not approved on the U.S. side. It's now apparently approved on the U.S. side. Who knows what's going to happen? So that's a possibility. And then the other major pipeline in Canada is what we call the Energy East pipeline. This one would actually go eastward and potentially having ports in Quebec around the Montreal area, or actually going out to St. John's in New Brunswick, which would be a port on the Bay of Fundy. So lots of different locations and lots of different waterways that these pipelines are crossing and interacting with. So the increasing amounts of dilbit that are moving through these pipelines has raised several questions. Because of the high viscosity and the high density of this product, the number one question we get is, will dilbit sink? So when it hits the water, will this product sink? Some of the other sort of questions that follow are, what will it do? Can we use our traditional mitigation techniques to actually deal with this product? Will it be biodegraded? And then if the answers to some of these questions are, it doesn't behave like normal crude oils, what can we do to actually adapt our mitigation strategies? So this is probably the most well-studied of the pipeline spills. This was in Kalamazoo, Michigan. And so here, there was a pipeline spill that was not detected for several hours, probably about 18 or so hours. So there was a delayed response. The oil actually didn't spill into water initially, but over the period of time, it moved across the sediment into a creek and eventually into the Kalamazoo River. And so what they're doing here is they're dredging up a lot of the stuff that did sink. And so this is where the will it sink question comes in. The challenge here is that, of course, in this instance, the oil interacted with sediment. And so you had this oil sediment slurry that ended up in the water and then it sank. So this is just sort of a highlight of some of the recent dilbit spills. And the unfortunate thing is that there's essentially very little or no evidence or scientific research that has been carried out for these spills. So I was just down at the Gulf of Mexico research conference, and that spill has been studied from top to bottom, left to right. Everything about that spill has been studied. These spills, very little has actually been studied. Kalamazoo is probably the best studied, but some of the rest of these, they didn't even, as far as I know, nothing's been published. So some of the ones from Canada, in 2007, there was actually a Sinbit spill from the pipeline that they're planning on twinning. And so what happened here was that somebody totally unrelated to the oil company was operating a backhoe, knocked the pipeline, and basically split it. And then the dilbit, or the Sinbit in this case, was moving through the sewer system, so they knew exactly where it was going to come out into the Burrard Inlet. They were able to set up booms before it actually got there, and they were able to clean up, they figure, about 80-some-odd% of it. So in the process of cleanup, they spent about $15 million, and that was probably Canadian, so it's a little cheaper. Mostly they did mechanical recovery. They did apply corrects at 9580 on shorelines, so that's a slightly different formulation than what was used in the Gulf. Kalamazoo River was more than 1 million liters. It moved down the creek into the sediments, and this one has, whoa, off we go. They spent over $1 billion cleaning this spill up, and that was over 2 years. It probably isn't actually still cleaned up. We still end up with moments where there's oil coming to the surface. Sometimes that oil isn't actually associated with the Kalamazoo pipeline spill. It's actually oil from other sources, but they're still doing cleanup there. 2013, Mayflower, Arkansas. I wanted to highlight this one because that's this picture here. It was actually 12,000 liters, so relatively small, but you can see that it just went straight through a residential neighborhood. So these pipelines are not necessarily off and away from where we're living. They can actually be very close to where we are. This one was actually a very rapid response because people saw that there was a leaking pipeline because it was right there. And so they were able to clean up most of it. However, the following year they actually went back, and then it kind of got back into these trees, so they were pulling out some of the sediment. More recently, last summer, there was about 225,000 liters of dilbit that was spilled into the North Saskatchewan River. Basically, there was heavy rains, the riverbank destabilized, the pipeline cracked, it dumped it right into the river, moved directly downstream, and they basically had to find new drinking water sources for 3 different communities because this stuff was moving rapidly that way. Lots and lots of riparian zone damage because of the cleanup, and that was also true in the Kalamazoo area. I believe at least 2 of these communities are still using their alternative sources. Husky says that they have cleaned it all up. There was absolutely no research done on that. So these 2 that I've starred here, Mayflower and Burnaby, those were the most successful cleanups. They were also the ones where there was very rapid response. And because of that rapid response, they were able to sort of minimize the coverage. These other ones where we did not have rapid response, there was significantly more oil released and then more of an impact. So dilbit, why do we care about dilbit? The 4 constituents of basically crude oils includes our saturates, our aromatics, resins, and asphaltenes. And what you can see here comparing the Arabian Light, the South Louisiana crude, to AWB and CLB is that while These light crudes have high amounts of saturates, so lots of alkanes, which lots of microbes can eat, and then relatively more aromatics, which again, a lot of those aromatics can be degraded by microbes. Our dilbits have significantly more resins and asphaltenes. So they tend to have things that are resistant to degradation, but we don't know what kind of an impact those kinds of compounds actually have on the environment. So also here is the density, and so when you're looking at the density of these guys, you can see that that's getting awfully close to the density of fresh water. So COOGER has been working to understand fate and behavior of dilbit and answer those questions, the big will dilbit sink question. I'm kind of focused more on what will the dilbit do in the ecosystems, what will be the impact, and then what will be the role of biodegradation in mitigating these spills. And so at COOGER, we have this very large wave tank facility. There's actually 2 tanks in there. One of them is a wave tank where they can generate waves with specific intensities and look at droplet formation, the impacts of adding things like Corexit, interactions, so they've done oil sediment interaction experiments. And then they also have a flume tank where they've carried out things like weathering studies. So that's the chemical side. So some of the work that they've been doing is looking at dispersants and then looking at sediment and looking at sediment and dispersants together. And this is just highlighting some of the work that the chemistry folks have been doing there. So here we have dispersant effectiveness, and this is Corexit 9500, and this is a different type of dispersant, SPC 1000. You can see that there are different interaction or different effectiveness of the dispersants. And what we have here is this is the orangey color is a winter blend, and this lighter blue is the summer blend. So you can see that the dispersants actually work differently depending on which blend we're dealing with. And Corexit was, generally speaking, far more effective than the SPC 1000. So this is looking at Corexit and suspended particulate matter and oil. This is just oil and particles, and then oil only. And so we've been characterizing the formation of these OSA, oil sediment aggregates, and understanding whether that can help to actually disperse the oil. So this is the interaction between particles and oil help disperse the oil. Does it just help sink the oil? And what are the mechanisms of formation? We have colleagues at the University of New Jersey who is actually modeling the interactions between the sediment and the particles. And I think what we're going to do, as he and I started talking this week, that particles and sediments are really cool, but particles, sediments, and microbes is even better. So we're going to start throwing the microbes in there. So we're looking at weathering. What part of the question of will dilbit sink has to do with how does dilbit weather. So we have a really light constituent, some sort of condensate, mixed with something that's really heavy. Do those, when they're mixed together, stay together and act as a single unit? Or do we have two things acting almost like a really light crude and then a really heavy crude? So how are they actually behaving? And so they've been doing some weathering studies. This is actually looking at BTEX being released over time from two different slicks that were different thicknesses. So we're trying to understand if you have a really fat slick, so fat at 7 millimeters, you actually have a much higher amount of BTEX released, which would make sense because you've got more oil in that slick. But it's actually released over 36 hours versus a thinner slick where you have much more rapid release of your BTEX. And we care about the BTEX because those are the benzenes, the toluenes, the really easily evaporated things that tend to have toxic impacts when we breathe it. Getting towards the does it sink question, this is density of weathered products over time. And so up here, this line represents our fresh water. And you can see that relatively rapidly, admittedly about 22 degrees Celsius, within about 2 to 3 days, we have densities in these products that are higher than fresh water. So the potential is that with very rapid weathering, you could have sinking in systems. The sin bit was always quite a bit lower. This was also at 12 degrees Celsius, so temperature can have a major impact on the weathering. So about 48 hours. So I only started with COOGER back in April, and they've been trying to address one question over the last few years, and then I'm going to introduce you to the new questions that we're going to try and address. And so the first question that they've really been focused on for the last little while is, can the in situ microbial community degrade dilbit? They started asking the question when they were working on the East Coast about whether the microbes that were in the North Atlantic could degrade the oil that they would be exposed to there. And then now we've moved to dilbit. This is being carried out using little microcosm experiments where they can add nutrients to maximize the response and look at whether the microbes can actually respond to just the dilbit itself. And as well, they were using Corexit 9500. And this is focused on the Douglas Channel region. This was in preparation for developing mitigation plans if the Northern Gateway Pipeline had been approved to go through Kitimat. So I'm going to show you data that answers this question, and then our new question is, what is the response of the microbial food web? So not just bacteria, we know bacteria like oil in some cases, but what about the rest of the organisms that are in that, the water column? And then can we predict, or in some instances, can we manipulate the response? Could we improve it? Could we use biostimulation by adding nutrients to increase the rates of degradation? And so this is going to be carried out using mesocosm experiments where we can monitor impacts of nutrients. We're not using dispersants right now because there's no sort of protocol for using dispersants on dilbit at this moment. And we're going to focus first on a coastal marine system because Bedford Basin is right outside my door and I can easily access it. And then we're actually going to move on to fresh brackish water systems. So the first question, are natural microbial communities able to respond and degrade bitumen? So we're here right now. This is where I work, and this is where the experiment was carried out. And we were interested in this because this was going to be the proposed ending of that Northern Gateway pipeline. There's also a proposed liquid natural gas terminal for this area. So Kitimat's here, so this is where the pipeline would come in. This would be the tanker traffic. There is an aluminum smelter there, and there is a shipping port already. So it's, as I say, it's a relatively pristine area, but by no means is it absolutely pristine. However, over here, this is the Haida Gwaii, and this whole bottom part of the island here is Guayhanas National Park. And so that's a very pristine area. It's also an area where we have a high amount of interest by the indigenous people who would like to be able to continue carrying out their fishing and maintain their culture, basically. And they're very dependent on sort of the ocean system for that culture. So in this area, there has actually been, basically this whole region in here, an informal tanker moratorium since 1972. And it was actually finalized in November. So part of the saying Northern Gateway Pipeline is dead. The moratorium was actually formalized. But it's a moratorium that excludes a lot of things. So you can't have crude oil tankers, but you can have boats, and you can have liquid natural gas, refined products, but of course that's what the tankers are carrying. They're also carrying bunker C oil and all of those other heavy types of oils. I do also want to point out that right in here is where the Queen of the North, which was a ferry, actually sank and ran aground. And so there's a ferry there that is leaking diesel fuel sort of slowly on a regular basis. But it is, as I say, it is sort of relatively pristine, a very nice location. So some of the concerns about the dilbit spill there would be high seasonal discharge from the Skeena River. So freshwater lens, that gets us back to that question about sinking and density. High particle loading, so oil mineral aggregates or all particle formation. It's relatively cold water, so we're talking about, it gets up to maybe 18 degrees Celsius. So unlike the Gulf, where we have 30 some odd degrees Celsius sometimes, it's relatively cold. And it can get down to about 4 to 6 degrees in the winter. And so that increases, of course, the viscosity of the oil and decreases the metabolism of the organisms who would be degrading it. It's also, it is a deep fjord system, and anytime you get freshwater lenses, deep fjords, you tend to end up with periodic low oxygen because you have good stratification, high organic matter input. And so the bottom of the fjords can actually go quite anoxic. And anaerobic degradation of oil products is really slow compared to aerobic degradation. So they set up these microcosms. So they used surface water from the area, so they went up on some ships and collected surface water and set up these microcosms in these baffle flasks where they added surface water, they added dilbit, they added nutrients, and then sometimes they added dispersants. They incubated them, they monitored oxygen over time using a little oxygen sensor, and then they would sacrifice them at different times to measure alkanes and PAHs, and then look at the diversity and activity of the microbial community. So I apologize for these graphs, they're a little bit full, but what you can see here is this is 2014, 2015 within the channel, so up in the fjord, and then this is actually 2014, 2015 out in the Hecate Strait there. And there's always these guys up here, these two lines, which is actually just saltwater and nutrients, and saltwater, nutrients, and dispersants. So the oxygen, this is actually measuring oxygen saturation. Oxygen was rarely reduced when we just added dispersant. Relative to these other ones down here, which is where the, in this case it was the Cold Lake blend was added. So as soon as oil was added, we pulled down the oxygen concentration, with the one exception of this right here, where this one actually had Cold Lake blend and we didn't get a response. So there was a different response in 2015 in Hecate Strait. What we don't know is whether that's biological response. So is the oxygen being pulled down because the microbes are eating the oil? Some of these, if you can see the green and the yellow, and especially in this one where the green and the yellow actually pull down the oxygen faster, that's with filtered seawater. So the microbes are essentially gone from that. So we could have some sort of either biologically mediated oxygenation of hydrocarbon species or some other sort of just chemical oxidation. They were in the dark, so we don't think it's strong light occurring here. What we did have was rapid degradation, or relatively rapid degradation of the alkanes. So here's our alkanes. These are half-lives here, so we have very short half-lives for the alkanes in essentially all of our experiments. However, these are our alkylated PAHs. You can see, so this one goes all the way to the top, is 347 days for the half-life. So alkylated PAHs were poorly degraded, and so were the PAHs, especially in this one treatment here. So this is no dispersant is the gray, with dispersant is the blue. And just to point out, dispersants did not always increase our degradation rates. In some cases, dispersants actually slowed down the degradation rates. So this was actually a theme, again, at the conference this week, was you'd get one person would stand up and give you one set of data, and then the next person stand up and say the opposite. So things are highly variable and can change quite a bit. So hydrocarbon degrading microbial communities did respond. I don't have the, all of the data right now. So this is actually what the natural community looks like. And if you guys know your microbial groups, this is the gamma proteobacteria. And these guys are the ones who responded. So whether there was oil by itself or oil with dispersant, these guys increased in abundance. And it was what I like to think of as the usual suspects, the ones that we always hear about. Alteromonas, Coelia, Pseudoalteromonas, Alcanoborax, and other Oceanosporiales. So the usual gamma proteobacteria that we see in the Gulf, we see in the Arctic, we see in the Atlantic coast, we see the same guys on the Pacific coast popping up and degrading. So we really answered the question, can the in situ community respond? The question is yes, they definitely can. So what controls their response? And how does dilbit affect other trophic levels, and can we augment it? And so I'm not actually going to look at these other trophic levels. So these are organisms that are found in the Strait of Georgia outside the Port of Vancouver. So these are guys who could potentially be exposed to dilbit. I'm going to focus on sort of the lower trophic levels, because if we have an impact that's affecting phytoplankton or zooplankton, these organisms are also going to be affected. And so it started with some preliminary experiments to try and get a handle on what might be controlling some of the responses. And in this case, we use seawater, weathered dilbit, and nutrients. But what I wanted to do was get away from the nutrients that had been used before. And I'll show you some of the data that shows just how much nutrients were being thrown in some of these experiments. And so we measured O2 consumption again, alkanes, pHs, but I also wanted to monitor the nutrient concentrations and the cell abundances. So I wanted to count the bacteria, see if they were growing, but I also wanted to see what happened to the nutrients. And so oxygen consumption was highest when we had dilbit. So what I have here are 4 different treatments. The lighter ones are without oil, so this is just straight seawater, so straight out of the Bedford Basin. The lighter blue here is when we added Bushnell haze media, which is a common media that's used for doing hydrocarbon degradation. It is about 300 micromoles of ammonium and nitrate and another 200 micromoles of phosphate, which is orders of magnitude higher than the nutrients you're going to find in the Bedford Basin. So really high nutrients. And then here we have with seawater with just the dilbit, and then seawater with the Bushnell haze added. This is just showing you the oxygen milligrams per mil over time. And you can see that there was a difference over time depending on whether dilbit was added or not. Essentially, there was no significant difference between with or without nutrients. It was only with or without the dilbit. So, and most of the oxygen you can see was actually consumed in the first 24 hours. So these are the consumption rates, much higher with dilbit in the first 24 hours, and then after 24 hours it was just, nothing was really happening. These are the nutrients, and I do want to point out that these are on log scales because that's the only way I can show you what's in the Bushnell haze concentrations, because they're so high. So nitrate and ammonium, so we were starting at about 5 micromole with nitrate. We then had, I think it was 380 micromoles, so just a little bit. And then ammonia was a little bit lower. At the starting point, it was a little bit higher in the basin. So at low concentrations, so if we just look at the seawater and the seawater with dilbit, at low concentrations, so this is T0, T96 again, the amount of nitrate that was used was about the same as the amount of ammonium that was used. So when they had low nitrogen available, they used both nitrate and ammonium. When they had really high concentrations of both available, they essentially only used the nitrate. So we had a decrease in the nitrate, and they essentially did not use ammonium. So they swapped what kind of nitrogen they were using depending on the concentration. Did that affect how they were actually able to degrade hydrocarbons? A little bit. So here we have the alkanes, the methylated pHs, and then the regular pHs. This is the cell abundance. So with nutrients, they grew better. Or with dilbit, they grew better, sorry. This is just seawater by itself. This is seawater with nutrients. They grew faster when the nutrients were present with the dilbit, but in both cases, with or without extra nutrients, they reached the same concentration. they did degrade alkanes faster. So we had, over 96 hours, we had significantly more alkanes degraded with the addition of nutrients. But essentially the other ones didn't change. And so couldn't we use this to actually increase degradation by manipulating nutrient additions? And the problem is, if we go back to this slide, is that if we have that much ammonium and nitrate left over after the bugs have finished doing their degradation, we have a different kind of pollution problem, right? So we might solve our oil pollution problem by introducing our nutrient pollution problem, which can then stimulate primary production. Things like harmful algal blooms could occur. You can end up with massive blooms sinking down anoxic areas. So we kind of shifted one problem and turned it into something else. So can we actually increase degradation, manipulate the nutrients, but not cause a problem for ourselves? And so in this case, I wanted to try and look at more environmentally reasonable concentrations of nutrients. So we actually collected seawater, and then we filtered it out and collected all the bacteria from the seawater and put it into artificial seawater that had really, really low concentrations of nutrients. And then we added in either nitrate or ammonium or phosphate, or combinations of nitrate, phosphate, and ammonium phosphate. And so this is where we started to see some interesting results. This is looking at oxygen concentrations here. And so basically in the control there was very little oxygen consumed. I do also want to point out that the temperatures that we did this at was between 2 degrees and 8 degrees Celsius. So we're actually doing this in a unit where we're trying to heat things up and stop things from freezing, because as we set it up, we got a cold snap. So we're trying to keep things a little warm, but it's still relatively low temperatures. So when we add in dilbit with any of the nutrients, so all of these nutrient additions also have dilbit added, we have drawdown of oxygen, which we'd seen before. But what we do see is that in 2 cases, phosphate alone, or ammonium with phosphate, we actually had significantly higher oxygen usage. So we did see a difference depending on what nutrient was added. And when we added them with nitrate and phosphate or ammonium phosphate, I tried to keep the ratios kind of similar to what we were seeing in the environment. So when it comes to growth rates, we also saw a little bit of a difference. Essentially, dilbit decreased growth rates for all of these guys. So this is the highest growth rates. This is cells per hour is in our control treatment. They don't show up super well here, but they were just going, and they were ready to keep growing and growing. Anytime we added dilbit, we had a reduction in the growth rate, except this ammonium plus phosphate, where we actually had slightly higher growth rates compared to the dilbit. So we did start to, again, see some different responses. How about degradation? Previously, we had done our analysis with GCMS, so we could get our alkanes and our alkylated pHs and our pHs. However, I did some calculations, and that represented approximately 0.05% of our total pHs. So our total petroleum hydrocarbons are not alkanes and pHs. They're mostly a lot of other stuff. Or they're higher, larger saturates and pHs than we're measuring. So what I did here is we actually did GC-FID for total petroleum hydrocarbons. So that's shown here. And then we did TLC FID analysis to look at the saturates, aromatics, resins, and asphaltenes. And what we see here is essentially no change over time, or very little change. There's a lot of sort of messy data here. So TPH concentration, even at T equals 0, where we added the same amount, at least we thought we were adding the same amount to every flask, we have highly variable numbers. And then, you can just see these error bars kind of all over the place over time. And then with the TLC-FID analysis, what we would expect to see would be a decrease in the percentage of saturates and then increase in the percentage of resins and asphaltenes. That one I actually have the new data. We had some samples that were, specific samples we were rerunning, but we don't really see a shift from t equals 0 to t equals 96. So we don't have clear detection of degradation. And so did it occur? Probably there was some degradation occurred as I did the calculations here. So the GCMS is measuring nanograms per liter versus the GC-FID where we're getting milligrams per liter. And dilbit itself just seems to be highly variable. So you take any different sample of dilbit and it doesn't necessarily look like the sample you just collected. But essentially, if we go back and we look at the GCMS data, where we had dilbit by itself or dilbit with BA, with nutrients added. With the nutrients, we were down to about 26% of the alkanes degrade were remaining at the end of the experiment, so we got rid of about 75% of the alkanes. But these other numbers are still pretty high, so we still had 84% of the methylated pHs and 52% of the pHs. And those, of course, are the ones that are considered toxic and a problem. So we don't have great degradation occurring in these systems. So we're going to scale up to from the microcosms to mesocosms. So the flume tank, they're using the wave tank a lot to do experiments. They're not using the flume tank. So I'm going to turn it into a giant bathtub for my experiments. I'm going to have a flow-through system where we can have 15 mesocosms set up. They'll have their own light source because we've got a roof over the facility. And we'll be able to either set up ANOVA type experiments with different treatments, or we could do regression style experiments. So this coming year, we've got 3 different experiments planned where we're going to do basically the nutrients with the dilbit and see what happens on a larger scale. And we're hoping that maybe we can start answering the question of will it sink under natural type conditions, and if it's just spilled onto the water surface. So we're going to have it all set up with, we'll have some loggers, we'll have some sort of sediment trap to try and pull anything that's falling to the bottom. We're using Teflon bag, and we'll have air hose and a sampling system. So it's a little bit based on a system that I used when I was down in Alabama, and we did some work with the Macondo type oil. So this will hopefully help us address some of these broader questions of how do other trophic levels respond, how much degradation can occur, what happens to the dilbit, where does it go, what happens in fresh water, and can we predict responses so that given certain environmental conditions, temperatures, nutrients and things like that, could we tell responders this is what you should expect? Because that's what they want to know. They want to know what to expect so that they can then plan appropriately. And so acknowledgements, as I say, I only got there last April, so a lot of this work is not mine. So these are people who work with COOGER at DFO. Sequencing, we work with a group at NRC, and they're actually based at McGill in Montreal. And then we started doing some work with the SOA over in China. And so I will take any questions that you might have.
Do you have sufficient data that you can calculate the kinetics of the biodegradation?
We're going to try and get that. It seems like most of the degradation really occurs in the first little while, so the first couple of days maybe, and then after that they kind of settle down. So we're going to try and sample sufficiently during that period of time that we'll have a good idea of what's going on, whether there's delays in the degradation or what's happening. Yep.
So in the beginning, how are they assessing cleanup? Because they're, you were mentioning they're basing like their efficacies on cleanup's percentage of a pure phase, or are they talking about residual saturation that's in the subsurface? They're just talking about the surface, free napple, or
so that's one of the things that isn't really well defined. And there was an excellent speaker at the meeting about the Gulf Coast who was talking about, the Coast Guard says that the tar balls are cleaned up to as close as possible to background levels, And his argument is that background was here, their levels are here, it's not even close, and is it really as close as possible? So the problem is that clean is never really defined, and so usually it comes down to a government agency and the people who caused the spill agree on what clean is, and they get to that point and they say, okay, we're done. So, unless somebody calls up and says there's an oil slick, come deal with it.
Right, because obviously as they infiltrate in, there's going to be different forms of it,
Yeah.
Whether it's free phase or whether it's residual saturation, and then you can have wettability issues. So were there any investigations on comparing the wettability of these dilbits versus, say, like a light crude? Because they'll,
I'm going to just tell you right away, I am the biologist in this program. So I don't know everything that they've been doing on the chemistry and sort of physics side of it. But a lot of that work is being done by different groups across Canada are working on that very kind of, those kinds of questions.
So could you show the slide where you show the density changes in the experiment?
The density. The weathering densities?
Yes. So does this experiment in a mesocosm or how does this experiment perform? What is the energy?
So for this particular experiment, they were using the flume tank. So they had it set up where they had a little ring that they poured the oil into. And for each oil, they had 2 rings. They poured the oil in, and then they started water flow. So there was a little bit of water flow in the system. And then they just sampled over time.
So, and do you have an idea of the depth of the flume?
So the whole tank is about 2 meters deep. And for these ones, so those are like 4 millimeter thin slicks that they were pouring.
And do you have any idea what the energy they use for mixing it?
I personally do not, but I know that they've got that.
The reason I ask this question because all this, the density effect,
Yeah.
it depends on the energy, how much energy involved.
Yes.
And also I think the BTEX that is used in a solid, you get the from dissolved concentration of the fluid. But if it is a particular phase, because when you have mixing the bitumen, you have two phases. One between the bulk phase and the
Yeah.
particular phase. The fate of particular phase depends on the size and also how this aggregate and density would depend on that one, because the aggregation of the 2 forces determine
Yeah.
the
So I think
density.
the density they measured based on collecting samples, and so what was left basically in the slick on the surface.
Slick on the surface.
Yes, I believe is what they were looking at. So in some of the experiments that they did, they saw little droplets actually coming off the bottom of the slick and kind of dropping down. So not sinking to the bottom, but droplets that would start to come off the bottom and kind of hang out about 5 centimeters below the surface or something like that.
So density on the surface slick.
This is density of the surface slick.
Even if we be more careful about the density in the water column, because that will determine the phase. Because whatever in the surface, they're going to be the surface. Because this density is lighter, that's why they stay in the surface. But if it's in the water column, if the density is high, density is getting higher, that is the same. But the
Yeah, I
surface
mean, there could be stuff that is sunk off the bottom of this. I think something like 70 to 80% of the weight remained in the slip. Something like that. Again, I did not do this work, so.
I think from my perspective, it seems like that the lighter one, density that is lighter to remain in the surface.
Yeah.
But we're more concerned about the density of the downs. And also, I think the size that will affect how to fade up. Thank you.
I have 2 questions. So the first question, may you show at the beginning of the slides, parameter called dispersion effectiveness. So manner and how is that assessed? The other question, may you
Should not have
show.
put any of that data up.
The other question, may you see the nutrient addition experiment?
Yeah.
Why nitrate is preferred than ammonia?
Okay, so the dispersant effectiveness I can send you the paper.
Yeah, so I can't read the paper, just wondering.
Yeah, I don't know. I did not do that.
I think I can answer that one.
Okay.
I think nitrate is more energetically like if there's a difference in nitrogen and ammonia, they go for nitrate. It's like in terms of the electrostatic energy, there's less energy to use the nitrate.
Nitrogen, hydrogen.
Nitrate than ammonia. Ammonia you need more energy.
Not always. Depending on the organism, they would prefer either one.
So some organisms can only use one or the other.
Yeah.
And
Depends
some
on what you
organisms
have.
will preferentially use one or the other depending on their situation.
Yep.
And so the difference is probably has to do with the identity of the organisms present in their physiology. And so that data, I'm waiting for the final experiment that we did where we added the nitrate or the ammonia or the phosphate. We're actually waiting for the 16S analysis and the nutrient analysis because I want to see what nutrients were left and whether they actually got limited or not. So.
Following this question, what are the natural levels in the system of nitrate, phosphate, and ammonia?
So the Bedford Basin ranges for nitrate, I think the average is about 5 micromoles, and the ammonium's a little bit, average
In
is a little bit
surface
higher.
water?
In surface water. Yeah, so we actually have a time series.
Mm-hmm.
So I went through and I pulled, well, somebody else had pulled all this data, but I looked for sort of the average concentration. So the experiment with the artificial seawater, the low nutrients was actually 2.5 micromoles of each nitrogen source and 0.04 micromoles of phosphate. So it was really low, and then we added in sort of like 10-fold higher. So it went
And
up to
they would
25.
actually switch depending which one is, in higher concentration and limiting, right?
Yeah, and so
That's what you're saying.
that's what we want to see is, do we have, so if there's nitrate available or there's ammonium available and they degrade oil, are the same organisms degrading the oil, or do we have totally different communities that respond?
Most likely different.
Yeah,
I don't know.
I mean, there might be some that are the same and then some that are different, so we'll find out.
Is not that affected by pH?
The pH probably doesn't change that much in these systems.
Water is buffer.
Yep.
You didn't mention the oxygen, dissolved oxygen that you were monitoring during the experiments. Did that change dramatically? And did you monitor trace element concentrations?
We didn't do trace elements. The oxygen didn't change very much at all.
Really?
Maybe 1 or so percent, and it dropped down right at the beginning, and then within 24, after the first 24 hours, it really didn't change very much. The challenge with the oxygen is that the system that we have, I'm pretty sure leaks enough and variably enough that the oxygen data is sort of more of a relative comparison. I'm pretty sure there's oxygen coming in as fast as there leak. So.
I might have missed the answer for Yvonne's question. So dispersant effectiveness?
That's the question I cannot
Yeah.
answer. I can answer the nitrogen one, but
Yeah,
Was
so
that by droplet size, isn't it?
it could have something to do with the droplet, because they've got a list that they put in the tank, and then they can measure droplet size distributions.
Because I mean, I'm sure they're affecting either solubility and or mobilization potential, right? From the water column. So I didn't understand how they were assessing which one was.
Yeah, it's just droplet size.
Yeah, so-
they're exposed for degradation.
Yeah, they've got, I think, 2 lists, which are particle size imagers that are in the tank at different distances away from where they have the slick, and they spray the dispersant. And then they can see what size and then the
Size
distribution.
Of the oil droplets?
Of the oil droplets, yeah, the distribution. So when they're doing those experiments, they get rid of all the annoying biology. So basically all that's left in the water column is oil droplets, so they can see those easily.
Does the use of dispersants affect the primary productivity of the system?
I don't know.
That's a very interesting question.
Judging from what I saw this week, the definitive answer is maybe. There was literally one talk saying diatoms get, totally reduced and they can't handle dispersants, and then the next talk says they're very resistant to dispersants. So, yeah, I don't know.
I think it's like two phenomena is working here. When you use the dispersant, the main intention is to reduce the size. And
Right.
if you reduce the size, you have the higher surface area, it
Right.
increases the degradation rate.
You
And
also
this-
increase the total pHs
Total pH.
into the water column, which is something that didn't get measured a lot. So you might start with the same amount of oil, but natural dispersion versus chemical dispersion is going to change your distributions.
How do you have like any suggestions that will support that degraded, like the dispersion actually affect the activities of microorganisms? Does it decrease their activity or something?
It decreases the activity of some organisms, but will increase the activity of others. Yeah.
There are a
So,
lot of papers on that on the Gulf,
yeah.
Deepwater Horizon.
Yeah. And from the microcosms that they did, there are specific organisms that tend to pop up when dispersants are used that don't pop up when you don't use the dispersants.
Any other questions?